Hi everyone, welcome to the Michael J. Fox Foundation's Hot Topics webinar today, focused on the importance of biomarkers research in Parkinson's disease research. My name is Mark Frazier. I'm the Vice President of Research Programs here at the Michael J. Fox Foundation, and we're really excited and thrilled to have this discussion on the importance of biomarkers and some of the state of the field of current Parkinson's biomarkers. I'd also like to thank Dr. Ken Merrick for joining us on today's panel. Dr. Merrick is President and Senior Scientist at the Institute for Neurodegenerative Disorders in New Haven, Connecticut. He's a member of the Fox Foundation Scientific Advisory Board and is principal investigator for the Fox Foundation's flagship biomarker study called the Parkinson's Progression Marker Initiative. So we're excited to have him today. I'd also like to thank and introduce Dave Iverson, who will be moderating today's discussion. Dave is the Fox Foundation's contributing editor and patient council member. Before turning it over to Dave, I'd just like to thank Merck for the sponsorship of the 2013 Hot Topics webinar series, and also highlight Merck's support for the Parkinson's Progression Marker Initiative, the biomarker study. So with that, I'll turn it over to Dave to lead this discussion.
All right, thank you, Mark, and thank you also, Ken, for being part of our discussion today, and thanks, everyone, for joining us. It's a really important topic, and while we so often know within the Parkinson's disease research field that there are so many ups and downs and successes and sometimes reversals of fortune, this is one where there is a lot of promising news and a lot that we can be optimistic about learning in the years ahead. So let's get started. We want to talk first about what an actual biomarker is, what it gives us, and one of the best ways, I think, to understand that is to look at some of these examples we have on the screen, because there are many, many diseases where there is a biomarker, where there is something that tells us what's going on with the disease. And we see these examples on the screen, from blood pressure to an enzyme test for liver function to a gene mutation that carries high risk for developing breast cancer. So Ken Merrick, let's start, if you would please, with the blood pressure one, because that's in so many ways the most obvious. Describe, by way of what we're looking for in Parkinson's disease, the analogy of a blood pressure cuff for hypertension.
Sure. Thanks so much, Dave. First, let me thank the Fox Foundation for hosting this webinar and on this very exciting topic. It's a pleasure to be here. Again, the way I think about biomarkers in general, as illustrated in this slide, is just an objective measure of disease. And of course, as a clinician, we seek objective measures to help us to further understand clinical symptoms and clinical progression. These are tools that help to really assist us as we understand the clinical symptoms that individuals with Parkinson's disease may show. So, of course, it would be great to have a simple measure like a blood pressure cuff that we could use to monitor whether individuals might be either developing Parkinson's disease or to monitor their symptoms as they progress. The big disadvantage we have in developing biomarkers for brain disorders is, of course, that the brain is sort of harder to get at. While it's relatively easy to take blood or to do the kinds of procedures that, like a blood pressure test, it's much harder to sort of test what's happening within the brain itself. And so, I think that we'd like to develop tools that can give us information about what's going on in the brain. These tools could be a range of different biomarkers, which we will discuss over the next few minutes, including ways to image the brain, to take pictures of the brain and watch the changes in nerve cells as they occur. as well as trying to acquire fluid from around the brain, or ideally even fluid in the blood that reflects what's happening in the brain. So, again, what we really seek are simple measures that could be utilized that would reflect, as blood pressure reflects heart disease or stroke risk, that would reflect what's happening in Parkinson's disease.
And Mark Frazier, one of the things that's interesting about looking at these examples from other diseases, which we don't have yet validated for Parkinson's, is that they tell us different things. A blood pressure cuff, for example, can measure what's going on at the current moment, can allow you to test whether or not a drug you take lowers your blood pressure or not. The gene example, made best known most recently, of course, by Angelina Jolie, is something different. It assesses your risk for developing a condition, and that's part of where we're going with this effort in Parkinson's as well, right? To both find markers that would identify a way to measure what's going on at a given moment with the disease, but also things that would identify the potential for someone to be at risk for Parkinson's.
Right, that's exactly right. So the BRCA gene may tell us a little bit more about who might be at risk for developing breast cancer. And similarly, there are some rare genes found in Parkinson's disease that are found in families that have a high prevalence of Parkinson's disease that can indicate one is at increased risk to developing Parkinson's disease. These genes are rare, as I said, in certain families, but they give us an insight to some of the underlying genetic causes of Parkinson's disease. And it is thought that most cases of Parkinson's disease are caused by a combination of genetic risk factors and also potential environmental risk factors. So the genetic understandings and these genes that are identified really give us a way to predict whether someone may develop Parkinson's disease later in life.
Well, let's look at where we're trying to go now with our effort in identifying and validating biomarkers in Parkinson's disease research. And some of the things that Ken and Mark have both just referenced, imaging and genes, we will get to what we're learning about those so far. But let's first kind of walk through the ways in which biomarkers can be useful, because they can actually be useful in a variety of different ways. And the first is what you're seeing on your screen now, which is to make it more precise in our diagnostic efforts, so that these will be a way we can really identify whether or not someone has Parkinson's, first of all. Because right now, Ken, Merrick, as many people listening in on this webinar know, the way in which your neurologist provides diagnosis is a pretty crude mechanism. It's that finger tapping kind of exercise, or watching whether or not your arm swings. We're wanting to find something that's much more precise than that, yeah?
Absolutely. So I think that clearly, Parkinson's disease remains a diagnosis which is established based on clinical symptoms, and that can be both subjective and often somewhat crude and difficult. So we seek to have more objective ways to assist physicians in making a more accurate and earlier diagnosis as well. And so one of the main roles of biomarkers is to try to provide physicians with more information at an earlier time to help them to be more able and more certain of diagnosis. So one of those ways, I'll go back to, I mentioned earlier, is an imaging tool which has now been recently approved by the FDA to assist in the diagnosis of Parkinson's disease. That's called DAT Scan. But there are, we'd like to develop simpler methods and additional methods to help to be certain earlier and about the diagnosis. This kind of goes back to what you were alluding to a moment ago, Dave, which is that there isn't one role for a biomarker, and the corollary to that is that it's very unlikely that there's going to be a single Parkinson's disease biomarker. There are going to be several, each of which are going to be utilized in somewhat different ways for diagnosis or to monitor progression or to monitor the response to therapy, as well as potentially other ways as well.
And these then also can, part of why this is important before we get to our examples, are in what this next bullet point on the slide, which is that they'll assist in how we select patients for clinical trials, because right now we're never entirely sure, in part because the diagnosis itself is uncertain. We may wind up with people in clinical trials who actually don't have Parkinson's, and that in turn affects the outcome of our clinical trial research. So it seems like a small thing, but it's actually quite big, because it will allow for clinical trials to proceed in a much more precise way.
No, again, this has become a very important issue, particularly as we seek to develop clinical trials that involve individuals at the earliest stages of Parkinson's disease, where the diagnosis may be the least accurate. So it is really crucial to have very accurate ways to ensure that the individuals enrolled in a clinical trial indeed have Parkinson's disease. I'm just going to imagine if, for example, something like 10 or 15 or 20% of the individuals enrolled in a study don't actually have the disease in question, it makes it very difficult to prove or disprove whether a drug is effective. And that situation is not so far-fetched. It actually has happened, and one of the things that we're doing in this study that Mark alluded to, originally called the Parkinson Progression Marker Initiative, is to utilize biomarkers to try to identify individuals accurately as early in disease as possible to, again, ensure we can ultimately use that approach in subsequent clinical trials.
And as you were also referencing, and Mark Frazier at the Foundation, why don't you chime in on this? We're going to be looking at several different ways to measure what's going on with the disease, including how the disease is progressing. So we might have one marker that tells us whether or not you have Parkinson's, and that assists the diagnosis. There might be something else that will tell us how the disease is progressing over time. Again, something that's going to be important in how we test new drug possibilities.
That's right, and really the need, one of the major needs in Parkinson's disease therapies is for new drugs that actually modify, slow, or stop the progression of Parkinson's disease. There are symptomatic drugs available to treat the symptoms of Parkinson's, but there is currently no medication available that really will slow the progression. And one of the challenges in developing new drugs that will slow the progression of the disease is that current clinical trials to test whether a drug may slow the progression of the disease still rely on the interpretation of the clinical symptoms by the neurologist that Dr. Merrick described. So it is somewhat of a subjective interpretation on whether the symptoms are getting worse, and it requires, because of this subjectivity, it requires a large number of people to participate in the trial. And because the Parkinson's disease is a slowly progressing disorder, it requires to follow these people in the clinical trials over a long period of time, 18 months to 2 years, to really understand whether this new intervention is changing the course or modifying the progression of the disease. So a real need to help and assist these clinical trials of new potential disease-modifying drugs is to have these objective markers that we know change with the course and the progression of the disorder, and ideally we know that we know they change in a shorter amount of time, so we can shorten the clinical trials, so that these objective markers that change with progression can be used to complement the clinical interpretation and the interpretation of the neurologist looking at the clinical symptoms to really have a better understanding, a more complete understanding of whether these new drugs are modifying the course of the progression, either slowing or stopping Parkinson's disease.
So maybe I can just add one more point, which I think is in addition to what Mark was just saying, is that with another important issue that we are have learned from monitoring clinical disease progression is of course something that every patient with Parkinson's disease already knows, is that there's a lot of heterogeneity in Parkinson's disease progression. Some people progress slower, some faster. And it would be very helpful as well if we identified biomarkers that would assist us in predicting how fast someone is going to progress. This may, of course, ultimately lead us to more targeted therapies as time goes on, but that's sort of another way in which biomarkers can be helpful in trying to sort of sort out the subsets within Parkinson's disease, so we can be more better and more accurate about understanding disease progression.
It's a really key point, and it brings us to the last part of this bullet point before we get into our examples, which is the way in which biomarkers can be useful for treating. Because part of what you're suggesting, Ken Merrick, is that while many of people listening on this call all have the same diagnosis, you have Parkinson's disease, within that diagnosis, there's tons of variation. And if we can identify biomarkers that will give us more clarity about those different subtypes, then we might know that person number 27 on this webinar would most benefit by a particular treatment, and person number 37 on this webinar might benefit by something else, right? It'll allow for perhaps a more personalized approach to how we treat people. Is that right?
Sure. No, that is exactly what we seek, and I think is what we expect will happen. And I think that's an area where biomarkers can be really are crucial to enable us to do this.
All right, let's start looking at what you're finding so far, because this is the really exciting work that both of you are engaged in, because we're already beginning to find some really interesting things through the work that you're engaged in. Let's start at the top with these different kinds of scans. You've referenced DAT scans, Ken Merrick, and we actually have a couple of people who have already written in some questions on that, so let me read those. And encourage everyone, by the way, that if you'd like to post a question to either Mark Frazier or Ken Merrick, to go ahead and fill that in. There's a screen in the upper right corner of your computer screen. Just type in your question there and get it to us, and we'll try to get to as many as we can over the next 35 minutes. But someone writes in, Ken, what have you learned from the DAT scans? I'm a participant in one of the studies.
Well, let me just take a step back and just, talk for a moment about what DAT scans are. So DAT scans are dopamine transporter imaging, is simply a way to assess the number and the integrity of a group of nerve cells in the brain that make this substance called dopamine, which we know is primarily affected in Parkinson's disease. So it's one way we have of sort of directly examining within the brain whether, to the extent to which the dopamine system is involved. And so it has provided us with a great deal of information about the course of Parkinson's disease, so understanding that by the time individuals develop early symptoms of Parkinson's disease already, from an imaging perspective, about 50% of the imaging activity is gone, is reduced. So there is likely a prodromal or premotor component of Parkinson's disease during which there is activity, there is disease within the brain that we can't detect with symptoms, but could be detected using imaging. And finally, and perhaps more importantly, on a day-to-day basis, I mean, imaging has become very valuable as a tool to assist in the accurate diagnosis of Parkinson's disease. You mentioned a little while ago about the need for accurate diagnosis in clinical trials, but in clinical trials now, when imaging is used, often in early Parkinson's disease, about 10 or 15% of individuals may be misdiagnosed, and imaging tends to be a more accurate way to identify whether there is a dopamine problem in the brain. So it has proven to be quite useful, but it certainly does not take the place of a clinical examination and clinical follow-up and clinical management.
But Ken, it would also seem that this is something that sort of validates the reality after someone has already lost a lot of their dopamine supply. So in terms of assessing risk or getting in early, it's not as helpful. Would that be fair?
Absolutely, yeah. I think that, again, it provides you a kind of a window of a couple years earlier, maybe 2 years, maybe 3 years earlier than would sort of the development of symptoms, but it is likely that people who've developed Parkinson's disease may have already indications within their brain 10 or 15 years earlier than that. And so we would really like to have even earlier markers of risk and of early neurologic brain degeneration beyond that we see with the current imaging tools.
And Mark Frazier, what differences are there between these various scans? We see on the screen DAT scan, PET scan, MRI. Someone writes in, one of our participants, how are MRI scans actually useful? I think of MRI as being used to, whether or not you've torn ligaments in your knee or whatever. So explain what the differences are between these various types of scans.
Sure. Well, DAT scan and PET scans can really help with understanding some of the specific cellular changes at the cellular level of the brain. So they really provide a window and a picture of certain types of brain cells that change. So, for example, DAT scan provides a picture of changes that occur in cells that make dopamine, and which are the main cells that are lost in Parkinson's disease. MRI imaging provides a more global picture of some of the structural changes that occur in Parkinson's disease or other neurodegenerative disorders. So if there are, for example, in Alzheimer's disease, there's a part of the brain that called the hippocampus that gets smaller in Alzheimer's disease compared to people without Alzheimer's disease. So MRIs can detect changes that in brain structures that either get larger or smaller. And also it can, in some of the more sophisticated MRI scans, it can provide some insight into how the different brain cells are connected to each other and how that changes with disease course.
And how practical is it from just a financial insurance standpoint? Do you know, Ken Merrick, about someone has just gotten diagnosed, let's say on this call, but they're a little unsure, they'd like a second opinion. Is a DAT scan now something that someone can go out and get, or a PET scan and have that diagnosis validated? Will insurance cover that? What do we know about that?
Yeah, so DAT scan is an approved diagnostic tool in the U.S. and in Europe. It is really designed for only a subset of individuals because for two reasons. One is that it is quite costly, and the second is that it tends, not to be absolutely necessary if the clinical symptoms are quite clear and the physician feels quite confident in the diagnosis. But there are certainly a subset of individuals who have symptoms which are not so clear, or who would like additional certainty. And it is, in general, possible to get a DAT scan. The insurance sort of landscape is varied from state to state as to how easily that is reimbursed and covered. but in general, it is available. But I think it would be important to be in touch with your physician to understand in your location what the insurance situation is before proceeding.
Let's move over, and we'll come back. If people have more questions, please do send them in to us about scans. But let's move to the question of biological indicators or potential biomarkers for Parkinson's. And Mark Fraser, this is one of the really interesting things that we're beginning to learn from the studies that are being launched and the PPMI, the Parkinson's Progression Initiative that the Foundation has supported in terms of what we're learning from spinal fluid, from cerebrospinal fluid. And that is that there's something showing up in that fluid that, at least in our early data, and I know it's only early, that's quite interesting. Describe what it is that you're finding within the fluid of people who have Parkinson's.
Sure. So we collect spinal fluid in a number of different studies, and the reason that we collect spinal fluid is that it's the fluid that bathes the brain. So it's closer to the brain than, say, a fluid like blood or urine. So we think that may provide some special insight to what is happening, what are the changes that are occurring in the brain. And interestingly, so these changes that I'll describe, I would say, are still in the research phase and earlier in the development than biomarkers such as DAT Scan, which has been FDA approved. So this is still early in development, but intriguing nonetheless. There is a protein called alpha-synuclein that is implicated in Parkinson's disease in a number of different ways. One of the ways is that it forms clumps in the brain cells of people with Parkinson's disease called Lewy bodies. And so alpha-synuclein is one of the major protein components of Lewy bodies. Additionally, we can detect the alpha-synuclein protein in spinal fluid of humans, and in several studies, including the PPMI study, we are seeing that alpha-synuclein protein in spinal fluid is significantly lower in people with Parkinson's disease than age and gender matched control, so lower than the control population. And this is interesting. The hypothesis, why is this lower? We don't really know at this time, but the hypothesis is that because alpha-synuclein accumulates in these Lewy bodies in the brains of people with Parkinson's disease, it is sort of sequestered in the brains, and that's why you see lower alpha-synuclein in the spinal fluid of people with Parkinson's.
And Mark, is this something then that could be used? We talked earlier about the ways in which these different biomarkers could be used in diagnosis, in the assessment for who could participate in clinical trials, and to test whether or not a particular drug is working for a particular person. Where would this fall, assessing someone's, because spinal tap's not something you want to do, every month or so necessarily. So where is this something diagnostic? Where does this fall?
Sure. So right now, the information that we have from the studies available is that it really distinguishes, it may be able to help distinguish a Parkinson's person from someone without Parkinson's. So it's really at the diagnostic level. What we don't know is how the alpha-synuclein level in spinal fluid changes with progression of the disease, and we hope to have that data, some of that data by the end of this year, at least some early insight into whether this biomarker could be utilized in understanding whether a drug affects the progression of the disease. So at this point, it is sort of a diagnostic. It may be a diagnostic tool. It may be useful as a marker of progression, but at this point, we don't know yet.
So in other words, while it might be a way to identify who has Parkinson's, if I take drug X, it wouldn't necessarily affect the alpha-synuclein level. That might be something that would be measured by some other marker?
That is a good question, and it's open for debate. So there are a number of drugs in development that are targeting alpha-synuclein as a therapeutic strategy, and there's a number of reasons why we think lowering alpha-synuclein in the brain may be beneficial and may be a disease-modifying therapy. So for those drugs that are actually targeting to lower alpha-synuclein, the spinal fluid levels of alpha-synuclein may be a very nice biomarker to understand whether when people taking this potential drug that lowers alpha-synuclein, looking at whether the drug affects spinal fluid levels, either increases or decreases, could be a very nice biomarker to understand whether the drug is actually affecting alpha-synuclein levels and would assist in the clinical trials and assist in the development of those drugs.
And one other question on this, and then we'll get to our other examples and start taking more of your questions. But Ken Merrick, as I understand the early results, something really interesting is also being assessed, which is that the levels of alpha-synuclein in the spinal fluid may also tell us something about these different subsets of Parkinson's, that people who have tremor-dominant Parkinson's have one level of alpha-synuclein, and people whose Parkinson's is more a problem of gait or balance might have another. Is that right? Tell us what you're finding out about that.
Right. So this goes back to the comments we made earlier that there are, of course, different kinds of Parkinson's symptoms within the broad umbrella of Parkinson's disease, and one way to divide that group is individuals who have predominantly tremor as their most important symptom, and while others don't have much tremor but have more difficulty with their balance and gait and so forth. And if we divide the group up by those categories, there does appear to be some difference that we are seeing in these very early data in their alpha-synuclein levels in their spinal fluid. So these are very encouraging and promising, and I think fortunately we will have much more data emerging from the PPMI study very soon that will either confirm this or give us more information or create more questions. But I think that type of data is exactly what we seek, and again, ultimately can feed back on those groups that have drugs that might target synuclein to understand, who best to target.
Here's a good question from someone who wrote in that has to do with this question of what we're learning from the spinal fluid through these proteins like alpha-synuclein and some of the others that you see listed up there, tau and beta amyloid. This person writes in, is there a predictive measure for dementia associated with Parkinson's based on alpha-synuclein, tau, or beta amyloid? Can you answer that, Ken Merrick?
Sure. No, that's a great question, and certainly the cognitive impairment is a major concern in Parkinson's disease, and is unfortunately a common component of Parkinson's disease. And so it's very helpful, or would be very helpful, to be able to understand who might be at greater risk for having cognitive impairment and greater severity of cognitive impairment. So they're sort of borrowing from our colleagues in the Alzheimer's world. There does appear to be a relationship in Alzheimer's disease between the levels of some spinal fluid proteins and risk of cognitive impairment, and we are very actively examining this in Parkinson's disease as well. I would say so far we don't have that answer, but we hope and expect that the collection of spinal fluid that's ongoing in PPMI will sort of shed light on that issue shortly. I think there are other ways to try to correlate with with spinal fluid assessment in trying to understand the risk of cognitive impairment. And this sort of brings us back to the fact that we need more than one biomarker. So at the same time, we are going to be looking at specific imaging markers that focus on cognitive impairment, as well as, of course, specific clinical tests that try to also provide us with insight as to whether individuals are developing cognitive impairment. Put all that together to try to come up with an index as to how whether we can understand who's going to be more at risk to develop cognitive impairment.
So do you imagine, Ken, in time that we'll have a kind of menu of tests and options so that you as a neurologist would be able to say to an incoming patient, we're going to run a bunch of different tests here, and that's going to tell us, A, whether definitively you have Parkinson's. B, it's going to be able to maybe tell us something more about the orientation of your condition and what you're at most risk for. And C, that's going to be able to tell us, what treatment we might want to suggest at this point, and then that we'll be able to monitor over time. I mean, it's putting together all of those different possibilities.
Yeah, that is the goal, and absolutely. And I think you mentioned personalized medicine earlier, and I think that this is really what we're, in a sense, what we're driving at is that within, again, the sort of Parkinson's disease umbrella, we would like to be much more specific about identifying ways in which a specific individual may change over time with regard to their Parkinson's disease and what therapies might be most beneficial for them, and of course to monitor that to ensure that that is correct and potentially modify the therapy as time goes on. That's the goal, and absolutely, and we're not there yet, but I think we are making some progress in that direction.
And Mark Fraser, we haven't really talked much about, we hinted a little bit earlier that there's some genetic connections to Parkinson's just as there are for the breast cancer risk gene, the BCRA gene. One of those in Parkinson's is LRRK2, LRRK2. And so that also is something, and part of what's interesting about this, I guess, is that it would be one of the ways you could get in early on. If someone carries that mutation, we know that's a risk factor. These are one of the ways, right, that we could maybe get in on the disease early on and potentially be able to stop it before it even starts.
That's right. I mean, that as you said, the LRRK2 mutation is relatively rare in terms of its occurrence in a general Parkinson's population. In certain ethnic groups, it does have a higher prevalence, but it identifies individuals potentially before symptoms progress or develop. And theoretically, one could initiate treatment earlier in the disease course. One of the ways that the Fox Foundation is addressing this is we have developed a consortia of clinical sites that have identified individuals that do carry some LRRK2 mutations, both people that have Parkinson's disease and people that have not developed Parkinson's disease, and really followed them clinically and collected information using brain imaging and collecting some biologic information, spinal fluid and blood, to really understand how these individuals develop Parkinson's disease. I should mention not everyone that has these mutations develop Parkinson's, so it's really an important question to understand those individuals that don't develop Parkinson's that have this mutation. How are they different than people that do develop Parkinson's? And these types of studies and these types of biomarkers will clearly contribute to our understanding of Parkinson's disease in general, but also identify markers that may be indicative of one that may develop symptoms that would then be a reason to potentially initiate therapy early.
So does that mean, Mark, that if someone, for example, tests that they have the LRRK2 mutation, and that's a test you can take now, and if in time we're able to understand why someone who has that mutation doesn't get Parkinson's, that there may be something else going on in that person. Maybe they have another gene that works to counteract that, and that could point the way towards a possible treatment, a way to sort of fix the LRRK2 problem?
That's exactly right. We are supporting several studies that are called genetic modifier studies, and it's what it really means is to look at individuals that maybe carry this LRRK2 mutation and do some genetic screening to identify other genes that may influence either developing Parkinson's disease or not developing Parkinson's disease. So one can imagine identifying a gene or several gene changes that are actually protective and do not are protective from developing Parkinson's disease. And in that sense, those would identify new drug targets and ways to potentially increase the gene products, those protective genes, in other individuals. So that kind of drifts from biomarkers to really identifying new drug targets, but they are interrelated, and I think both types of studies are really useful.
We have a number of questions coming in, Ken Merrick, about blood tests. One person writes, I've heard that there are new blood tests that may soon be available for pre-screening. Someone else asks, are there validated blood biomarkers? And yet another person says, can these same things that you're finding in the spinal fluid also be found in blood? So bring us up to date, if you can, about what may be identifiable within blood tests for Parkinson's.
Certainly, we would love to have a blood test for Parkinson's disease. Of course, it's so much simpler and easier for everyone to be tested in that way than in spinal fluid. Unfortunately, at the moment, there are no blood tests that have been useful for diagnosis or following progression of Parkinson's disease. Part of the problem, again, is I think what we discussed earlier, that since Parkinson's disease, of course, is a disease of the brain, the sort of the most proximate fluid is spinal fluid, and that's the one, unfortunately, which is the most likely to give us information. Blood is much less likely, but still, as I think someone asked in their question, there are those same proteins that we have identified in the brain in blood, but it's harder to know whether they are coming from other parts of the body or from the brain or both, and it gets therefore difficult to understand what changes in blood with regard to those proteins actually mean. So I think there is a lot of effort now being put forth to try to understand whether we can identify a reasonable blood marker. Ultimately, I expect that will happen, but at the moment that has not yet happened. And so, I think our focus on spinal fluid is that it can provide us with real information that can be useful today, but it doesn't mean that we're not continuing to explore other opportunities in blood, which would be easier.
And before I take our next participant question, Ken, if you would just briefly touch on what's in the lower left side of on our screens, which is olfactory issues and sleep disorders, because these are things that are happening right now, and these are both things which we think are early signals.
Yeah, so I think one of the real exciting findings in Parkinson's disease during the past few years is that we realize now, as many patients have realized for years, that it doesn't only cause problems with motor symptoms, with tremor or rigidity or slowness of movements, but there are other problems that arise, and some of them are outside the brain. One of them, interestingly, is that almost all patients with Parkinson's disease also have a loss of smell. People may not realize this, but if tested, it's very evident. And interestingly, that loss of smell may occur in advance of the other symptoms. Similarly, there are unusual symptoms related to sleep problems that can occur often in advance of Parkinson's symptoms. One of them is called REM behavior disorder. That's a funny sleep disorder that some people get in which they essentially kind of act out their dreams at night, and most of the time that's noticed by their bed partner. There are other problems that occur like constipation or bladder dysfunction that may also occur in advance sometimes of motor symptoms. So one of the issues that really has come up is, can we take advantage of these symptoms to try to identify, again, who might be at risk for Parkinson's disease, and ultimately utilize these symptoms as ways to initiate therapy at an earlier stage. So that's an ongoing process. Unfortunately, many of these symptoms are nonspecific, so they may occur in Parkinson's disease, but they may also occur in many other disorders. So I think that there are a number of studies now that are ongoing to try to better characterize how these types of early markers of disease might be useful in, again, identifying individuals with Parkinson's disease and ultimately sort of directing therapy at an earlier stage.
And it would also suggest the importance of family practice and internists and other members of your profession be aware of some of those, right? Because if someone has sense of smell problems, they're not going to go see a neurologist.
Absolutely. So again, it does. One of the important correlates of this is educating the general practitioners, internists about these additional features of Parkinson's disease, so that they might be thinking along these lines. Absolutely.
Let's get to more of your questions. Here's one, Mark, for you. Since it's unlikely that you're saying that a single biomarker will adequately serve the entire Parkinson's population, given the heterogeneity across patients, all the differences there are across patients, how many do you think it will take?
Yeah, that's a good question, and I don't know that I have a good answer for that. I think it will be more than one, and I think it will be a combination of both imaging and biologic markers and rely on a lot of the clinical examination. I think a key to kind of what has already been said is that there certainly will be different markers for different purposes. So you could envision several, maybe 2 to 3 biomarkers being used in testing new drugs to really understand whether a drug actually is benefiting Parkinson's disease and helping Parkinson's and slowing the progression. But those markers probably or may not be used in the management of that drug or of those of treating Parkinson's disease. So I think it will probably be multiple different markers for multiple different purposes, but I have no idea how many ultimately it will take.
Here's someone who'd like to know whether or not Merrick ultrasound could be used as a way to get in on early detection.
Right. So ultrasound is a very interesting approach that has been used primarily, actually, by our European colleagues, but also now in the U.S. To begin to to provide an additional tool that might be an early measure of abnormality in Parkinson's disease.
What would it show? What would ultrasound be able to show?
So the way this works is that and most people are familiar with ultrasound, from the perspective of looking at unborn children, and it's the same kind of idea. So an ultrasound machine is this time pressed up against an individual's temple and their head, and those the ultrasound then tries to identify sort of the density of the material in the very small area of the brain where there are dopamine neurons called the substantia nigra. The substantia nigra is a tiny little part of the brain. It's called the substantia nigra because it looks black when you look at the brain. And you can actually detect this reasonably well if you're skilled and develop skills in doing this process. And what happens in individuals with Parkinson's disease is that that ultrasound measure does change in the majority of individuals with Parkinson's disease. So you can detect a difference between Parkinson's patients and individuals who don't have Parkinson's disease. It's not 100%, but it has been useful in the hands of a very well-trained ultrasound examiner. So that is another tool that can be used. It is more of a screening tool, so it kind of gives you a sense of abnormality. It doesn't seem, unfortunately, to change very much or change discernibly over time, so it's not so good as a progression tool, but it may be of some value, and particularly in those centers where there is someone who has been well trained in this particular technique.
And Ken, when you say not so useful as a progression tool, meaning it wouldn't be something that you could use it wouldn't be a way you could measure the effectiveness of new drugs, right? Or whether or not a treatment was working in the way of...
Correct. Yeah, it doesn't seem to be very useful in that way. It seems more that it might be useful as a way of, again, diagnosis rather than progression. Right.
Here's an interesting question to either of you. Could a positive reading of aluminum in a sample of hair, in other words, tissue mineral hair analysis, be useful or be considered a biomarker?
Well, I can start. So metals, hard metals, aluminum, iron, magnesium, have all been implicated in some ways in potentially developing Parkinson's disease. So those individuals, so there have been studies that have demonstrated that individuals that are exposed to high amounts of metals have a higher rate or higher incidence or are at higher risk of developing Parkinson's disease. So, for example, welders seem to have a higher prevalence of Parkinson's disease than the general population. As far as aluminum and hair, I'm not sure whether that would be a specific biomarker that would be unique to Parkinson's disease. Some of the challenges here is that metals have been linked to a number of different disorders, both neurodegenerative disorders and non-neurodegenerative disorders. So it may not be specific to Parkinson's disease, but if one was exposed to high amounts of metals in their lifetime, that may be suggesting that you are at a higher risk of developing Parkinson's.
We just have time for a few more questions, so let me see if I can get through as many as we can. Here's a good one on whether or not there might be any potential biomarkers in the gut. And that may get us back to the question of alpha-synuclein, I think, Ken Merrick, because we know that alpha-synuclein appears in a variety of places, including within the GI system.
Yeah, so this is a very interesting new finding. That we can detect, or it appears to be possible to detect alpha-synuclein in the sort of the nerve cells that control the motion of the gut, and so that brings us to a lot of interesting conclusions. One is, of course, that Parkinson's disease certainly may now affect not only the central nervous system, but the nervous system outside the brain. The other is that it may be feasible to use detection of alpha-synuclein in the gut as a diagnostic tool by doing a colonoscopy or a sigmoidoscopy, and it provides us with a lot of important information, both from a sort of potential biomarker perspective, but also kind of understanding the disease beginning as well.
Just have time for this last question that someone raises, which I think is a great last question for us to close on, which is how close are we? I mentioned at the beginning that this is one avenue of Parkinson's research where there's a lot of optimism. I think both of you feel that there's a really good chance that we are indeed going to be able to find validated biomarkers for Parkinson's. So how close are we? And as part of your answer to that, maybe also indicate why that would be so helpful for everyone listening on this call. Ken?
Well, this is always a very difficult question because, as I'm sure people on the call are well aware, all research takes much too much time and it moves slowly. But having said that, I do think that we do have some very promising leads in the biomarker area that can, I think, bear fruit in the next couple of years, and maybe sooner. As with all scientific discovery, it's unlikely there's going to be some giant breakthrough, and more likely that we're going to see constant improvement over time. And that's exactly what we are seeing, for example, with regard to our understanding of our ability to detect synuclein or improve our imaging agents. And this is really important because for a number of reasons. I'll just mention two. One is because I think we need to be able to very accurately diagnose Parkinson's disease, but we also need to be able to push back that diagnosis to as early in the disease as we can, both because we want to understand Parkinson's disease and because likely drugs may be even more effective if we can start them at the earliest stage possible. So I think it's really important to have a real sort of stable of biomarkers we can use to be able to move those therapies backwards in time.
And with that, I want to thank both Ken Merrick and Mark Frazier for their participation in our webinar today. And I'll hand it back to you, Mark, for your last thoughts on this topic and to bring our session to a close.
Yeah, I mean, I think Ken explained or summarized my feelings pretty well in terms I think we're a couple of years away from some significant progress on developing some well-validated biomarkers for Parkinson's that would really accelerate clinical trial development of some of these new potential disease-modifying trials. So I'm very optimistic about the progress that we're making in the biomarkers arena. I know we are at the end of the hour, so I'd like to thank Dave Iverson for moderating this discussion, and thank you all for joining and offering some fantastic questions today. I also wanted to remind you that our next Hot Topics webinar will be on Tuesday, September 24th at 12 Eastern, and the topic of that webinar will be genetics and Parkinson's disease. So we look forward to speaking with you and talking to you then. And again, thank you for joining. Bye-bye.
Bye-bye now.